So today we will talk about endomitosis. But before talking about endomitosis, we must know what is mitosis. What is mitosis? Mitosis is the cell division. So at the time of the cell division, cell has different stages when the cell divides. So the first stage is the G0 stage where the cell remains active, metabolically active, and after that G1 stage is there where the cell takes the decision that yes, it has to divide. And thereafter S phase. S phase is the synthesis phase. So in the case of the S phase, whatever chromosomes are there, it doubles in the quantity. Now we are telling that the chromosomes are present in the homologous form. So all the chromosomes are present in this form, so their replication occurs into the S phase of the mitosis. Means the chromosome doubles. So after the replication, the chromosome will look like this. So this is the normal mitosis process. In the S phase, whatever genetic material is present in the cell, it completely doubles, it completely duplicates. That's why we can look this type of the chromosomes, the double chromosome, one centromere which is bound with the two chromatids, right? So what happened at when the chromosomal division occurs? So in the case of the metaphase, all the chromosome they ties them into the spindle fiber. These are the spindle fibers. So what happens? The spindle fiber takes its position on the centromere. It holds the centromere. Now this is the moment thereafter the chromosomes are going to separate into the anaphase. So in anaphase what happens? Both the spindle fibers move in opposite direction towards the poles and it will take chromosomes with them. So this is the normal mitosis process. But what happens in endomitosis? Endomitosis is not like the mitosis in that the chromosome will keep on replicating, but cell division will not occur. Now, what will happen if it will continuously duplicate and the cell will not divide? So all the chromatid will be bound to a single centromere and they will split longitudinally. So here separation will not occur because cell division is not happening. So no anaphase, no metaphase, nothing is happening. In the S phase only, the cell chromosome is dividing multiple times. So for that reason, the chromosome look like thread. And this becomes a very giant chromosome. Now why this is happening? Why chromosome is dividing so many times but the cell is not dividing? Because it happens most of the time into the small organism, we can say the microscopic organism. It occurs in the other organism also, but where the cell division does not require, means all the chromosomes are not very important. Say example, if it is the larva, so larva, we know the habit of the larva, they are the voracious feeder. So what they required the digestive enzymes. So only the enzymes are much required. So all the chromosomes duplication and all is not possible. So they are not required. So whatever chromosomes or whatever enzymes are required, that particular chromosome only duplicates. Why it is duplicating? Because what is the function of this chromosome? This chromosome, we know they comprise of the genes, right? So these genes have the DNA, and this DNA, we know they have the process of transcription, translation, and thereafter we are getting the protein, and these proteins are actually the main thing which is required here. So for getting more and more and more quantity of the protein, we required more and more number of the genes, so that very fast transcription and translation will take place, and we are getting the abundant quantity of the protein. So for that particular reason, in the developmental stages of certain organisms, such giant chromosomes are being formed. And now we will talk about little bit its structure. So when we talk about its structure, as I told you, these are very big, this becomes giant. Now see, instead of two copies, they are two chromatids, they are making so many, so definitely they are larger. So I have showed only few chromatids here. They form 5 arms, 6 arms also. So generally these chromosomes are 50 to 200 times larger than the normal chromosome. So it's a big number, 50 to 200 times. So definitely it is going to enlarge the size also. When the size will enlarges, definitely its dimension will also increase, and it becomes very very prominent when you will observe it into the microscope. Now when we talk about the DNA, so definitely it will have the 1,000 times of more DNA. So endomitosis happens, and it forms the two type of the chromosome, which we all are going to learn in the detail. Now what we all are going to learn about this chromosome, Polytene chromosome and Lamp chromosome, we all are going to study how they were discovered and what is their structure, and thereafter we will be talking about its significance. So first let us talk about the Polytene chromosome. Polytene chromosome, it was first reported by the Balbiani. Balbiani is the name of the scientist who has discovered this chromosome. And then thereafter, now where it is found, I told you that we are going to talk who has discovered and where it has been found. So the Balbiani, when he was studying the salivary gland of the chironomous larva, he found. But most of the study shows they were first found into the salivary gland of the Drosophila. So actually, Balbiani has not given the name. The name was given by the name Polytene chromosome was given by the Collar, is the another scientist, and he was also studying the salivary gland of the this Drosophila. So when he studied, he found that it is not only restricted to chironomous larva or the salivary gland of the Drosophila, but it is also present in the cells of the rectal epithelium as well as into the Malpighian tubules in the insect. Rectal epithelium means the epithelial layer of the rectum, and Malpighian tubules are the part of the excretory system of the insects. They are functioning just like the kidney in the insect. So this Malpighian tubule also comprise, in short we can say, they are part of the digestive system of various insects. Now when we talk about Drosophila Polytene chromosome, it was found that it has 4 arms, and these arms have the alternate bands with the interband region. Now these alternate bands, when they were stained by the fusion staining technique, they found that this alternate band stains very dark and the interband region stains very light. Now why? Because the bands, they have the high DNA content. Now if DNA content is more, that means it has the more and more nucleic acid, so it will stain very dark, as we have learned into the heterochromatin and euchromatin, that the heterochromatin, the more DNA, more darkness will come. Now whenever, if any region which is absorbing more and more stain, definitely it will absorb more and more UV light also. So interband region which is staining light, so it will absorb, it will have the low DNA content and it will absorb the less UV light. Now when we talk about the bands, it's special, it exhibits a special swelling and this swelling is due to the maximum DNA. Now, maximum DNA means the maximum DNA activity is happening. Now we know the gene expression requires lots of machinery over there. That means the transcriptional machinery, lots of ribosome will be there. So the active region of this DNA, they will puff because of heavy load of the ribosomes, or you can say the transcriptional machinery. Continuous transcription is happening there. So for that particular reason, you will find the swelling in those particular region, and this chromosomal puff is named with the name of the discoverer, that is Balbiani. So these chromosomal puffs are also called as the Balbiani rings. So when we observe this chromosomal puff carefully, you will find that high RNA synthesis is happening. Now, why high RNA synthesis is happening? More and more RNA synthesis, more and more proteins will get. Now we will study a little detail about the significance of Balbiani rings. When we observe bands at a certain region, as I had told you that these bands, because of the high gene expression, they get the puffs, and these puffs are called as the chromosomal puffs. Now, when we observe carefully, you will find that these puffs are called, are having the large amount of mRNA, and these are pronounced during the molting. Now, why during the molting? What is the reason? Because the molting is the time when the development of the insect occurs. Now, insects have the different level of the development. They have the larval stage, they have the this pupal stage, so complete metamorphosis is there. So at the time of the molting, they have the various larval forms. So all the larval forms, they are just molting. Molting means they are shedding the previous skin, and that have a particular term in the zoology called as ecdysis. Ecdysis means removal of the previous skin, and the ecdysone genes, the ecdysone inducer genes A and B, they are continuously working. So for the formation of the ecdysone hormone, now all your hormones are also protein. So this ecdysone hormone formation requires fast gene activity, gene expression. So for that particular reason, you will find that at the time of ecdysis, these particular rings are pronounced, and this also can be seen into the salivary glands. Now, why salivary gland? Because these larvae, they are voracious feeder. So if they are feeding so much, so definitely they require lots of digestive enzyme, and these enzymes are again protein. So for that particular reason, you will find that they comprise of this Balbiani ring. So this is all about the polytene chromosome. Now we will talk about the second type, that is the lamp brush chromosome. Now this is again the second type of the giant chromosome. Well, it is called as lamp brush because when you will observe its structure, you will find that it looks like a brush which were used to clean the chimney of kerosene lamp. Hence its name is the lamp brush chromosome. Now when we talk about where it found, so it is found into the oocyte of the fish, as well as it is found into the amphibians, reptiles, and the birds. Now, who has discovered? Although it has discovered by the Walter Fleming, when he was studying the amphibian, but the detail he could not study. He just found that he has some lamb brush chromosome like structure. Sorry, some lamb brush chromosome is found, but its detailed study was given by J. Rooker when he was studying the oocyte of the shark. Now you will find it is also present in few of the invertebrates as well as plant, but their size is not as so much big. The biggest size lamb brush chromosome can be found into the salamander oocytes. It's having length of 5,900 microns, and it is found that they have the elastic nature. Let us talk about the structure of the lamb brush chromosome. So when you will observe them under the electron microscope, you will find that it has the two axes. One is the main axis and another is this middle one. What you can see is the main axis, and you can also find the lateral loop, which is also called as the loop axis. So when we talk about the main axis, so it has the main role. That means it contains the two chromatid which is rich in DNA. But if you will see this tip, so these tips do not have the axis or loop lateral loops. Why? Because these tips are just like telomere region. Telomere regions we have studied. These are the end point of the DNA. So these two tips will not contain the loops. Instead, this will have the telomeres. So when we talk about the region, certain region you will find that it gets coiled to form chromosome which are found into the pair. Here one pair I have shown in the enlarged form. So, what happened when you will observe under the electron microscope, you will find that these loops are of two types. Now, typical loop which will bend towards the central axis and it will give rise to RNA fibril. And this fibril are progressively increasing in length. One side of loop you will find is thicker than the other. Then second type of loop you will find are of special type. They are having the symmetrical region with the granules at the end of the fibril. Here you can see. The axial fiber is made up of DNA, but it is covered with a matrix-rich RNA and protein. Now, where it is present into the oocyte. Now, why oocyte? What's so special into the oocyte? When we talk about its significance, we know that oocyte requires the synthesis of large amount of yolk. If the yolk will not be deposited, then how the developing embryo will get its nutrition? So this high amount of yolk to be deposited into the egg, which requires the higher RNA activities and the protein synthesis. So this is all about your Lambrus chromosome and Polytene chromosome. A small quick review we will do. First, we have learned the endomitosis. Endomitosis is the process in which the chromosome divides but cell do not divide, and this makes the cell big in size. That's why these cells are called as the giant chromosome. We learned the two type, Polytene chromosome and Lambrus chromosome. Polytene matlab thread like structure. It was reported by the Balbiani, and he found that these are having the band and alternate band, interband and alternate band region. The band region is dark after staining, and interband is light due to the less quantity of the DNA, and the special swelling of the band due to the high RNA synthesis is called as the Balbiani rings, and this is responsible for the ecdysis. Ecdysis is the process of shedding the skin, and also it is present into the salivary gland enzyme. Then thereafter we learned about the Lambrus chromosome, which is usually found into the oocytes. The reason is now we have learned its structure, that it is having two axis, main axis and the loop axis, and the tips do not have loops. It just act as a telomeres, and these loops are of two type. And significance, we have learned that it requires for the large amount of synthesis of the yolk into the oocytes for the higher RNA activity as well as the protein synthesis. So at this note, we'll stop now.